How to determine the order of chemical reactions in a living cell? In what order does the
Смотрите
formation of
на
biological
последнее решение.
complexes occur?
Смотрите на реакцию
We use
на
classical
известных
thermodynamic
американских
theory to
политиков,
determine
которые
the order
прямо
of
говорят о
formation
том,
of a
что
hexameric complex.
э,
This presentation is devoted to the study of the assembly of a hexameric complex consisting of 3 proteins and 3 peptides. We will study the order of hexamer assembly by analyzing the calculated thermodynamic parameters. Let's consider the structure and functions of proteins.
Marburg virus encodes a nucleoprotein to encapsulate its genome by oligomerization and form a ribonucleoprotein complex. The VP35 peptide chaperoned nucleoprotein core remains in a monomeric state and completely loses its affinity for single-stranded RNA. Thus, the table presents experimental values of the binding affinity of the nucleoprotein and peptide, with various mutations in the peptides. Let us consider various variants of the formation of a hexameric complex, taking into account intermediate transitional complexes. Each such biological formation has its own set of physical parameters. The change in these physical parameters as the hexamer is formed can provide a forecast of the order of formation. This diagram shows all possible ways of hexamer formation for proteins of this type. We see the entire set of possible ways, including transition complexes. Each such complex has its own set of calculated physical parameters. In this case, the stability parameter and the logarithm of the dissociation constant. The next slide shows the experimental results of the dissociation constant for different mutations in the peptides during the formation of the hexameric complex. How can we explain such a difference in certain experimental values? The left side of this diagram contains the numerical results and schematics obtained for hexamers with higher affinity, compared to the diagrams on the right side of the figure, which present the numerical values and assembly order of the hexamer with lower affinity between biological subunits. Let us consider in more detail the obtained numerical results for the interaction of wild-type proteins and the V10D mutation in the peptide. The most stable biological formation in the upper figure is precisely the hexamer, which corresponds to the lowest values of stability and dissociation constant. Such a final equilibrium molecular complex is indicated in light green. So in the upper figure A there is a dead-end zone. If as the hexamer is formed, complex number 3.3 is formed, then the formation process ends here. We also see the codirection of the arrows for various calculated physical parameters. What do we observe when introducing a mutation into a peptide? Let's analyze the 2 lower calculated graphs that correspond to the V10D mutation in a similar way. Note the final point. For all 3 selected quantities, this is a hexamer. Also note the codirection of the arrows for each calculated physical quantity. The free energy value is maximum for the protein-peptide molecular formation, and the minimum value corresponds to a hexamer. Let's analyze the remaining mutations in the peptide, the introduction of which into the hexamer is characterized by good affinity. These are 2 mutations in the peptide M7P and Q9A. Let's first note all the numerical cases that achieve the set goals, either the minimum or the maximum in the case of entropy, for achieving hexamerization. Let's mark them with a cursor. As for the free energy, we see that the hexamer can be a kind of transition complex. In such cases the free energy values for the hexamer occupy intermediate values. Let us now return to the entire calculation scheme, containing both mutations that improve affinity and those that reduce the affinity of molecular blocks between themselves when assembling into a hexamer. Let us note the final molecular blocks for each scheme. As we see, in the right part, this is often the very initial molecular complex protein-peptide, that is, hexamers in the right part tend to disintegrate into simple molecular blocks. On the right side there is a case where hexamers break down into simple molecular blocks.
In this way, you can obtain physical data in advance for all possible simple molecular formations as the hexamer is formed and simulate the best and most advantageous chemical pathways when modifying the polymer chain. Contact our specialists with any questions. We will be happy to contact you and provide a free consultation on rationalizing a biochemical experiment.